Hi, and thank you for tuning in to my first lecture on Chapter 8, Mitosis and the Cell Cycle. In this first video, I'm going to give you an introduction to mitosis. We're going to cover chromosome numbers, and then lastly, chromosome structure. So Chapter 8 is all about cell division, which is really the process of one cell reproducing itself into two daughter cells. So there's two main types of cell division out there, and those are mitosis and meiosis. Here in this first half of Chapter 8, we're just going to focus on the first type of cell division, mitosis. And that really involves a cell basically making two clones of itself, or two identical copies of itself. So, my focus is going to be on multicellular eukaryotic organisms that are sexually reproducing, and that pretty much includes all plants, all animals, and all funguses. So everything I say here applies to all of those kingdoms of life. So the first thing I want to do is I want to take the time to define mitosis, and it's a form of cell division in which a parent cell divides in half into two genetically identical daughter cells. So the two cells that will result from mitosis will be identical to each other and also identical to the original parent cell. So what does it mean to be genetically identical? Well, that just basically means that if two cells are genetically identical to one another, that means those two cells have the same number of chromosomes, pretty much the same genome, the same genetic material. So that's kind of the overarching goal of mitosis, is to make sure that the two resulting cells have the same number of chromosomes as the original parent cell that we started out with. So I'm going to show you that here, just to give you like a broad overview of mitosis. I have a drawing on the right, and we're going to pretend we have a parent cell that's really simple. We're going to say, instead of it being like a human cell, which have 46 chromosomes, we're going to say it's just a cell that just has one chromosome in total, and that's it. So I'm going to draw that one chromosome here in green. Again, this is the parent cell. And one thing that parent cell would do prior to undergoing mitosis would be to replicate all of its genetic material. Now, this simple cell only has one chromosome, so it only has to replicate one of its chromosomes. If you're a human, you have to do this process to all 46 of your chromosomes. But what happens is that cell makes an exact replica of the chromosome, and it actually physically conjoins those two identical copies of that one chromosome. So the reason a cell has to replicate its DNA before doing mitosis is that cell needs to have 2 copies of its chromosome, one to give to each new daughter cell. So really what happens during mitosis is all you do is you take that duplicated chromosome and you line it up accurately and then pull apart the 2 copies of that chromosome and give 1 copy to each daughter cell. And the result will be something like this. And you'll notice that each of the 2 daughter cells look identical to one another and identical to the original parent cell. So if we're talking humans, human parent cells have 46 chromosomes, so we want to make sure that the daughter cells also each have 46. So I'm going to write out the primary goal, and we just said that. It is for So the goal is for each daughter cell to inherit a single copy of each chromosome. Collectively, a single copy of the entire genome. So our next question here at the top is, well, why does a multicellular eukaryotic organism like a plant or an animal need to undergo mitosis in the first place? Why would you ever need to reproduce more cells, like going from 1 cell to 2 to 4 to 8? Well, the primary reason is for growth and development. So think about this. Every plant, every animal starts out is just a single cell called a fertilized egg. And for humans, over the course of 18 years, you grow and develop into a multicellular adult composed of over 40 trillion cells. So how do you go from 1 cell to 40 trillion? Well, the answer is many, many consecutive rounds of mitosis, going from 1 to 2 to 4 to 8 to 16, and so on. So that's the major reason that multicellular organisms do mitosis, is for all growth and development. But then also, adults still undergo mitosis quite a bit. Like right now, millions of cells in your body are currently undergoing mitosis. That is because many of your tissues need just normal replacement, normal maintenance. Like for instance, the cells in your body, all your tissues, they have different lifespans. So for instance, like a red blood cell has an average lifespan of a couple months. A liver cell, like 6 months to a year. Epithelial cells, like those of your skin and those that line your internal organs, since they get damaged quite often, they have lifespans of like 2 days. So when I say lifespan, that means like every 2 days, one of your skin cells has to reproduce itself into 2 new daughter skin cells. So I'm going to write out just normal tissue replacement, also known as just normal tissue maintenance. And then third, if you have an injury, to repair that damaged tissue and grow new tissue, that is mitosis. So we'll write repairing injured tissues. So on this first slide, there's no new information. I just define mitosis again and also define what it means for two cells to be genetically identical. You can see a picture here at the bottom of an actual embryo of a marine worm going from 2 cells to 4 cells. It's like a time lapse from left to right. So next, I want to give a name to the cells in your body, or that of a plant or any other animal or a fungus. Okay, so the cells in your body that actually have the potential to do mitosis. So the specific cells in your body that at some point or another undergo mitosis are called somatic cells. So somatic cells are most of the cells of your body. That includes most of your 200 different types of tissues. So we can say, your liver cells, your skin cells, your heart cells, the cells of your eye, muscle cells, immune system cells, everything except for what we call germline cells. So germline cells are the one type of cell in your body that does not undergo mitosis. It's not that they don't divide, they just use a different form of cell division called meiosis. And we'll talk about that in unit number 3, which is the second half of chapter 8. So what are germline cells? So germline cells are the cells that give rise to sperm and eggs, and sperm and eggs are known as gametes. So sperm and eggs and the germline cells that give rise to them do not undergo mitosis. But every other cell in your body, every other tissue, does. And since they do, they're called somatic cells. Now, there are some exceptions. For instance, many cells of your nervous system and some cells of your heart, they actually don't divide at all. So you only get one copy of them. But most of your somatic cells do undergo mitosis at some frequency. Here again, we see the 3 major reasons that large multicellular eukaryotic organisms like ourselves undergo mitosis. And then at the bottom, I state again the primary goal of mitosis, regardless of what you're using it for. The goal is always for the daughter cells to receive 1 copy of each chromosome. So that's the primary goal. So our next section here, probably the most important section of this video, is titled Chromosome Numbers. So we're going to talk about 3 things: the number of types of chromosomes that you have, the total number of chromosomes that you have in a cell, and then also how many sets of chromosomes do your cells have. Now, since we're talking about mitosis here in the first half of chapter 8, all of these numbers that I'm going to take you through are with respect to a somatic cell. So we're not talking germline cells, we're not talking gametes. Their numbers are a little bit different. We're strictly talking somatic cells. And since we're humans, we're going to focus on human somatic cells. So the first question I want to ask is, how many types of chromosomes do your somatic cells have? Another word for a type of chromosome is a homologous pair. I'm going to draw something. Now the answer to my question is, humans have 23 types of chromosomes. They have 23 homologous pairs. So for instance, all of us, since we're the same species, have human chromosome number 1. It's the largest of the 23. And for all of us, it's pretty much the same size, same shape. Not only that, but human chromosome number 1 for all humans has the same genes located at the same spot on the chromosome. So you might ask yourself, if all humans have the same genes at the same location, why don't we all look identical to one another? And the answer is, all sexually reproducing populations have genetic variation. So there are slightly different alternate versions of genes in a population. And we'll talk all about variation and genetics and sexual reproduction in unit number 3. But we do all have the same genes. So what I'm drawing here is, this is supposed to represent human chromosome number 1. This next one, slightly smaller, will represent human chromosome number 2. And if we're talking humans, it would go all the way to 23. And you'll notice for each type of human chromosome that there is, you have 2 copies of it. You have one copy, which I've drawn in blue, called the paternal copy from your dad, and a second copy of human chromosome number 1, which I've drawn in pink, the maternal copy from your mom. So for each type of chromosome, you have 2 copies of it, one from dad, one from mom. So before I forget, I want to write the answer to this question on the left. 23 types, or you can say 23 homologous pairs. And that's what I was just starting to allude to. So for each type, you have a pair, and they're the same as one another in terms of their shape, their size, and the genes that they have. So that's why we call them homologous, like the same. Now, you don't necessarily have the same versions of those genes on your maternal and your paternal chromosomes, but they do carry the same genes. So I want to take the time to go to my slides and look at something called a karyotype, which is a stained image of an individual's chromosomes. So you can see here the 23 types. So here's type 1, again, it's the biggest. Here's type 2, 3, 4, all the way down to the smallest one, 22. And then a specially unique 23rd pair called the sex chromosomes. So this would be a male because they have X and Y. A female would be XX. And like I said, for each type, if we just look at chromosome number 1, you have 2 copies, one from the mom, one from the dad. Now, the second question I want to ask is, how many chromosomes does a human somatic cell have in total? And the answer to that would be 46. Now, each species kind of has its own unique number of chromosome types and a total number. And a bigger number doesn't necessarily mean you're a smarter, more complex organism. So for instance, I believe dogs have 78 chromosomes in total, 39 types, you know? Because some chromosomes can be bigger, some can be smaller. The number doesn't have anything to do with anything, really. For instance, onions have 14 in total and 7 types. I just want you to realize it's like a species to species thing. But for humans, we have 46 in total, 23 types. So again, if we go back to this karyotype, you can see all 46. Here's number 1, 2, 3, 4, 5, 6, and so on. Now, the goal of mitosis, which is our primary focus here in the first half of chapter 8, is to take all 46 of these chromosomes, line them up at the center of the cell, and then pull apart and separate the two identical copies of each chromosome and give one copy to each daughter cell. So keep that in mind. So the last chromosome number question we need to answer is, how many sets of chromosomes do we have? How many sets of chromosomes do human somatic cells have? And that answer is not unique to humans. It's not even unique to animals. This number is pretty much shared by most sexually reproducing organisms, which includes most plants, animals, and funguses. And since these types of organisms are sexually reproducing, it implies that they have two parents, which means you have two sets of chromosomes. So the answer to this question, how many sets of chromosomes do human somatic cells have? The answer is 2. You have one set from the mom called the maternal set. Okay, so you have a maternal set of 23. And you also have a second paternal set of 23. So 2 sets. And we have a specific term for describing a cell that has 2 sets of chromosomes. And that term is diploid. Di like for 2. So diploid is any cell that has 2 sets of chromosomes. So all of your somatic cells that undergo mitosis are diploid. They have 2 sets of chromosomes for a total of 46. Now I want to take the time to define this other term because we will use this term in unit number 3 when we're going over meiosis. Because some cells in your body, like germline cells and gametes, are not diploid. They're actually what we call haploid. Haploid like for half. And these are cells that only have 1 set of chromosomes. And it's important to produce what we call gametes that are haploid, that have 1 set of chromosomes, because sexually reproducing organisms need 2 gametes, a sperm and an egg, to fuse and to combine their genetic information. So you can't have 2 cells combining that each have 46 chromosomes, because then each offspring would just double in chromosomes, and that would be a mess. So you have to cut it in half, the chromosome number and the number of sets, prior to sexual reproduction, to keep our chromosome numbers constant as a species. But all I want you to realize at this point is that haploid means a cell that only has 1 set of chromosomes. So that's it. Those are all very important. I'll likely test you on all 3 of those types of numbers. So really important to know moving forward. So this slide here, no new information. We just covered all of this in my notes. I want to say one more thing about chromosome numbers. We have a way of writing this out and summarizing all of this information in one little equation. So for a human somatic cell, we would write it out this way. 2n equals 46. And if you do the math, n comes out to 23. So that is to say, human somatic cells have 23 types of chromosomes, so that's what n stands for. And we have 2 sets of 23 for a total of 46. So I just have this here. I'll periodically, just for fun, bring up Ancestry DNA and 23andMe. So these are results from me taking the test. So I've kind of combined results from 23andMe and Ancestry DNA. So on the left here, just kind of cool, you can see these are representing my 23 different types of chromosomes. Those are the 23 homologous pairs. And you can see for each type, I have 2 copies. One from mom, one from dad. And what they've done here is they've sequenced my genetic information, looking for specific nucleotides called markers that can give you all types of information, like in this case, ethnic breakdown. Markers can also tell you the likelihood that you're a carrier for a certain genetic disease, or maybe the likelihood of having a certain trait. But just interesting because you can get all of this information from the 46 chromosomes. So our next topic is chromosome structure. Now this is also pretty important. So before a cell undergoes mitosis, like I said, one of the major preparatory events for mitosis is to take all of your chromosomes and to duplicate them or replicate them. But before you do that, before you replicate your chromosomes, they're what we call unduplicated chromosomes, like I've drawn on the left. And it's just one double-stranded chromosome. So if I were to zoom in on this, it's just an extremely long strand of DNA. So I'm just going to make this up, but right? And that would go on for a while. As we learned in chapter 3, chromosomes have a lot of nucleotides, right? It's like millions of nucleotides in a row in one long chain. But my point is, before you replicate your DNA, you only have one copy of this chromosome. Now I want to show you what a chromosome looks like after you replicate it in preparation for mitosis. Oops. So after undergoing DNA replication, you no longer have what we call an unduplicated chromosome. It's now called a duplicated chromosome. Now I want you to realize, both on the left and on the right, they're both just a single chromosome. Okay? You don't need a duplicated copy. You don't need 2 copies of it. The only reason you duplicate it is so you have 1 copy to give to each daughter cell. So we have another name for a duplicated chromosome. It means the same thing. We also call it a pair. A pair of sister chromatids. Sister implying that they're identical to one another. So after you replicate your DNA, you don't just have one double-stranded chromosome, you have 2 identical copies of it. So for instance, right, this was A, I'm just going to keep it the same. you don't just have one of them. You have two identical copies. And once we finish mitosis, we're going to give one of each of these copies to each of the new cells. So these sister chromatids, not only are they identical to one another, they're also physically conjoined and attached to one another. So I'm going to draw that with these little dots. There are proteins that physically conjoin them and keep them together. So it's very important as a cell gets ready to do mitosis that it has all of its chromosomes in their duplicated form. So you can give a single copy to each daughter cell. So you can see all this same information here on the PowerPoint slides, same idea, nothing new. Then over here we have an actual picture on the far right of a chromosome in its duplicated form, and you can see the two sister chromatids, identical copies of one another, physically attached. And this will be our last slide in this video. We're going to go over two structures, two terms related to chromosome structure. And those two terms are kinetochore and centromere. So I'm going to use this drawing of a chromosome in its duplicated form to illustrate these two terms. So the first term I'm going to cover is called the centromere. And it's not an object, it's actually just a region along the chromosome. And it's the region where the two sister chromatids are most closely conjoined and attached to one another. And if you're looking at a picture of a real chromosome, it kind of looks like the waist of the chromosome. So right there is the centromere. Right at that constricted, narrow waist. Now, if you go back to that karyotype, you can see that each type of human chromosome has a centromere at a distinct location. Like, for instance, chromosome number 1, it's right in the center. For chromosome number 4, it's more towards the top. But that position is pretty much the same for all of us. So located at the centromere region is my second term. And I'm going to take a moment to draw that. So there are two of these, one on each sister chromatid, and they're facing away from one another. And these pink structures are called kinetochores. They are complexes of proteins that are attached to each sister chromatid. And they play a very important role in mitosis. It is these kinetochore protein complexes that help us pull apart the two sister chromatids during mitosis. So first I'm going to label it. And then I want to kind of draw out what their function is going to be during mitosis. And what's going to happen is cytoskeletal protein filaments called microtubules, which I'm drawing here in orange, are going to attach to the kinetochore of each sister chromatid. And they're basically going to pull in opposite directions and pull apart the two sister chromatids from one another. So here in orange, these are strong fibrous proteins called microtubule filaments, which we talked a little bit about in chapter 4. And my last thing to do is to define the kinetochore. And then lastly, these kinetochores will attach to microtubule filaments during mitosis. And again, what they're going to do is basically they're going to apply some force and help us separate those two sister chromatids, which again is the overall goal of mitosis. And that's it for my first video on chapter 8.